I'm Siddharth Pratap from Meharry Medical College, and I'm going to talk to you today about BioDIGS, Biodiversity and Informatics for Genomics Scholars. Disclosures, I am the External Consultant Committee co-chair for NHGRI's Anvil project. Michael Schatz from Hopkins, who is the PI of this project, is a scientific advisory board member for Cosmos ID, and Rita Caldwell is the co-founder of Cosmos ID and on our advocacy board for the Genomic Data Science Research Community Network. So this project is funded by NHGRI, and we get this question a lot. We're human. What are you guys doing sequencing soil? Well, soil is likely the home to the majority of all species on Earth. 59% of all life it exists in soil, and making it the most biodiverse habitat on the planet. So the group that I'm involved with, it is called the Genomic Data Science Community Network, the GDSCN, and we are a group of about 25 institutions, all smaller, historically black colleges and universities, HBCUs like mine, Meharry, minority-serving institutions, Hispanic-serving institutions, tribal colleges and universities, First Nation, and community colleges. Coast to coast, Puerto Rico and Hawaii are also members. And the common thread being that at underserved institutions, resources are limited, and faculty is limited in terms of how much commitment is there to training, the infrastructure, and many things. So, what does it take to get to critical mass to pool our resources and our availability to have genomic data science as part of our training curriculums, as part of developing the next generation of biomedical researchers? It's known that 16% of the U.S. is a Hispanic Latinx population. Maybe 9% of the genomic data science workforce is Latin Hispanic, 13% Black African American, and under 8% of the genomic data science workforce is Black and African American. So our focus is to train those next generation of folks. Okay. This is a paper from 2022, a perspective in genome research, where we outlined a roadmap to get to that level. This is going to take involvement of R1 research institutions to provide resources, lessons learned. At the center hierarchy of this is students who benefit from the collaborations between the R1 institutions and the underrepresented institution faculty, who are going to need release time from teaching and resources from the admins and the funders. So making this unique support system able to function. So within that framework, we're choosing to do this BioDIGS project as a pilot project to connect the participants. This touches on multiple levels of research. So for the microbiology and metagenomics folks, it's about discovery. What is in the soil? How do genomic compositions change in time and space and geography? For the bioinformatics folks and the genomics folks, how to optimizing metagenome assembly and short read versus long read functionalities for development. For agriculture and environmental, and a lot of the GDSC schools focus on environmental impacts. How do you characterize the soil and the microbiome in terms of like plant and animal development for agriculture? And on a larger level for public health, how does the microbiome influence human health and health outcomes? So all four of these tiers are places to explore. Okay. So the sampling protocol is outlined here, where we will apply for a permit or permissions to do the digs. Most of our sites are rural, suburban. There are some very interesting sites. Spelman College in Atlanta did it at a Superfund site because they're super interested in the environmental effects of that. So we will gather and pool the soil, take photos and GPS track coordinates of it. And this is repeated at 5 sites within each site's location. There are 15 of them so far, and this started at Hopkins, and these are the sites outlined here I'm on the Nashville site. The soil testing is done in parallel with the DNA sequencing, and analysis happens on platforms that can be agnostic, but we're doing them on Galaxy, Anvil, and Bioconductor or our studio. We find that we have a lot of diversity in terms of how our education members want to train their students, who are mostly coming from biology backgrounds or doing associate's degrees. So there's a lot of variation in how we choose to do the training. But so we wanted to be broad in that. Okay. So I'll hit three areas to outline here. The first is genomic diversity, second, environmental associations, and third, human health and disease. So on the genomic diversity side, the pilot data that I'm showing you today came from the Johns Hopkins sites. There are two counties that were sampled: Baltimore County with 24 samples and Montgomery County with another 24. With the geo tagging, we're able to get precise locations of this. And this was mostly done at a recreational site and also a wooded site at the Hopkins area. For sampling and sequencing, we're using Zymo DNA RNA Shield, which is good for 6 months, and that's super important when we're shipping from multiple sites across the country. QIAGEN DNA Easy Power Soil Kit for isolation, and then 2x150 on the NovaSeq S+. This gave 20 million reads per site. And for soil testing, we're looking at heavy metals like arsenic and cadmium, chromium, and also other members of the soil which are relevant to agricultural applications. Looking at the taxonomic classification, here we're using Kraken 2, and the vast majority of stuff we see is bacteria and protobacteria and actinidobacteria. Of high prevalence in the soil are nitrobacteriaceae, which are very common and involved in fixing nitrogen, as you'd expect. What else is observed is things that are relevant to human health, like streptomyces, naturally occurring antimicrobials, and members of the probiotic species like Verrucomic, I don't know how to say it, but things that are involved in like gut microbiome and probiotics. Looking at the assemblies here, we're looking at dark matter for microbiomes. Of all the assemblies that we ran, only 12 to 15% of them align to anything in the reference genome databases. 85% of it is unknown or unclassified. There were also some viral and archaeal samples, but we're kind of optimizing this for microbial. But that's another area of interest, is to look at the nematodes and the archaea and bacteriophages in there. But at this pass, it is mostly bacterial and microbial. But that 85% that is uncharacterized is super interesting for discovery. Looking at the order in clade level here, we're using SILF, which is using average nucleotide identities. It's 10 times faster than Kraken, which was necessary when we're trying to scaling to 20 different sites, and less memory intensive. So this gives us a pretty decent snapshot at the taxonomic level for all the sites, and this is what we want to do to implement going forward here. So I'll move on to environmental associations, the second phase. Looking here for heavy metal concentrations in Baltimore and Montgomery County, counties in the Maryland area, the soil sampling was used to look for iron, lead, arsenic, and multiple other metals. Looking at the order level and class, clade level, on the lead, this is the Vermont limit. It's different from state to state, and there's not a national limit on lead, but Vermont is 30 milligrams per kilogram. And a lot of the orders and clades are in soils that exceed the limit. Here we're looking at arsenic, and it's interesting, there's this bifurcation where there are several orders and classes that are in soils that exceed arsenic limits by EU standards. This is early data, but looking at calcium and boron and aluminum, there are definitely trends that are seeming to come with different orders and classes being more prevalent or less prevalent in different soil conditions. The diversity that we're observing in the geographic locations will be used to correlate to the soil sampling this way, and also looking to see, like, longitudinally what changes over time, whether it's seasonal or if there's a construction site somewhere close by, if that involves some changes going on. So enter longer reads. Okay. So we did Oxford Nanopore-Promethean sequencing for the Baltimore sites, and the total yield was 70 gigs with an N50 of 5.7 kbs, which was helpful. The short reads were needing help. So we did this in the SQL 2e, and this was done in the Baltimore area with that data, and it was somewhat helpful, but not terribly. And then enter the Revio. So this was very useful in progressing this project going forward. Out of like the 10 million reads, total yield was 85 gigs and an N50 of 9.6 kb. So very long curve on the large config size. That enabled, this is a cumulative distribution function, where on the y is the config length and on the x is the cumulative sequence length. So Illumina and SQL are here, and the Oxford and the Revio gave a very, very intriguing long tail. Up here with the large contigs are complete genomes. Okay, a lot of these, mags may be undiscovered, and directly below there are ones that are easy to bin and easy to join. So when we're talking about distributing this to the different schools for training, these become very compelling areas to send out, some data to one of the sites and have their undergrads work it up at a genomic level. And then this very intriguing long tail, where novel genes and plasmids and most importantly, low abundant species, which were not resolved with the short reads, would be discovered here. So every one of these could be, a cool undergrad project, a interesting summer internship, and there's hundreds of them to do. So PacBio came into the picture specifically here, and we should thank Dan Portek for optimizing the pipeline for analysis. Out of the 158 mags that came, 55 were high quality. Only one of them was assigned to a known species. Okay. Archaea were highly represented, but this gave us 25 times more high quality mags than the short reads. So order of magnitude improvement in there. And 27 single contig genomes came out of this pipeline and the SQL and the Revio sequencing. Okay. So finally, I'll do human health and disease. We're comparing platforms here as well. So we're looking at number of antimicrobial resistance genes found. And again, with the Revio, we had over 160 genes found, about 90 with the Nanopore, and lower numbers with the SQL and the Illumina. When we're looking at breaking down like what type of resistance, vancomycin seems to be the most common antimicrobial resistance gene seen in the Baltimore samples. It'll be also done at the other sites to look for that. My particular interest is looking at antimicrobial resistance, not in plasmid form, but in the form of where it's like genomically integrated into the bacteria. That hops around on islands and resistance islands and cassettes that are transposon mediated. If you look at one of the soil bacteria that's common, Acinetobacter, it's got several large insertion sequence elements in there, ranging from 10 to 100 kb. And the history of that, they come from Enterobacter and Pseudomonas from multiple different places. So, like bacterial gene transfer is directly relevant to some of the use cases that we'll do going forward. Within the vancomycin resistance, again, training level opportunities to get really deep into the granularity of what type of microbial resistance genes exist in particular sites population. Okay, so to the training. This is some of the materials that were created by Liz Humphreys and Ava Hoffman from Fred Hutch. This is the BioDIGS in the Classroom book that was created. I like it because there's a section for instructors, how to install or how to get going on Anvil, how to set up your classroom activity. There's a guide for students as well. So most of us are training undergrads or graduate students who are new to genomic data science. So these are super helpful in getting onboarded. And even if we're coming from different places on how we teach them coding, we use RStudio. Some other places are doing Python. This is a good entry point for analysis of applied data that is real. We find the students will be much more engaged when they actually dug up the soil samples themselves and they can analyze it here in next phase. So here it's one of our training results. So this is from Ment Ayalu's lab at Spelman College. It's a historically black college in Atlanta, all women. This is the Galaxy workflow that was created. And we're using Fly for assembly and Kraken for classification. This was implemented in her second year undergrad biology and microbial genetics class to undergrad level students. So Catherine Ulbricht and Nia Davis presented their implementation and finding of the Galaxy workflow at the Plants and Animal Genome Conference in San Diego, September of last year, and did a wonderful job. They also took this to Spelman Research Day, which is an annual day where everyone, almost all the undergrad students participate, and they won first and second place in the biology category. So really well done job there. Cool. And in terms of collaboration, so I want to highlight Dr. Kristen Reinhardt. She's with North Carolina A&T, Agriculture and Technical, in Winston-Salem, and we met her through this GDSC. Even though we're both at historically black colleges and universities, NCA&T tends to be on the engineering and comp sci focus, and we tend to be on the biomedical sciences side at Meharry. So I met Kristen through this network, which was just fantastic. She's running a program called Foundations for Grads 4C, which is serving as a hub for genomic data science training to schools like NCA&T and Meharry, smaller schools that are non-R1 institutions. Okay, so in summary, hopefully we've convinced you that exploring interactions between the soil and metagenomics is relevant to human health, not only for discovery of new genes and genomes, but for discovery of new antimicrobial resistance genes and islands. And orders of magnitude improvement are observed from long reads. So on the technology side, this seems to be a good use case. On the training side, to empower the next generation of scientists, teaching current genomics techniques in a way that is accessible is paramount to the function. So by providing like open access data, compute resources, removing hesitation to get on the clouds, at some of our universities. Next steps would be to like apply this in a dynamic space, like looking at longitudinal data, looking at changes over periods of time, and developing training materials for this to implement it into varied curricula, but with the same focus of training underrepresented minorities. Cool. I want to highlight the students who did all of the digging from all of the sites. And some of these students will be going on into the analysis phase of it. Want to shout out my PhD student, Lincoln, who is going to move forward in doing the analysis part for the bioinformatics in this stage. And I'll roll the credits for the PIs and the PDs and the folks who were involved at the leadership level for this. Again, Mike Schatz is the PI from Hopkins, and I've highlighted the work of Ment and Kristen. There are many other faculty as part of this network who have stories that are very compelling and very intriguing. Finally, I will acknowledge the funders and the GVSian faculty, Hopkins as the coordinating center, and the Fred Hutch Cancer Center for the creation of training materials. NHGRI funds our parent project, and I want to thank PacBio for optimizing the protocols and giving lots of in-kind sequencing or discounted sequencing to let us see how compelling the long read could be to complete this project. With that, I will thank you and take some questions.